Hello everyone and welcome back for some more biotechnology. In this chapter, we are really not going to present a whole lot of new information, but rather we are going to provide some context on principles and assays and techniques that we've talked about before, and we're going to really talk about them in the terms of biotechnology. So, ultimately, we've talked about this before, but one of the most important purposes of any biotechnology is to make human life better. And oftentimes that is going to involve developing medical treatments that will either diagnose, cure, or in some way alleviate disease. So, diseases, as we understand by now, can be caused by foreign pathogens, like what you're seeing here for meningitis causing bacteria, or we've talked about diseases like sickle cell anemia that are caused by genetic defects. We're going to talk about cystic fibrosis, another genetic disease later. Or we can talk about disease in the context of just normal breakdowns in physiological mechanisms over time, what we understand to be the aging process. So, a lot of the focus of medical biotech companies is going to be congruent with developing treatment and eradicating eventually human disease, including some of the following. And now this is certainly not an exhaustive list, but this should give you a pretty good idea of what we're trying to accomplish with medical biotechnology. Number one, this is mostly academic in nature, but we want to determine the cause of human diseases, and that's going to make the treatment a lot easier. So a lab that focuses on something like this is going to ask questions like, what is the genetic basis of this disease? Is it caused by a mutation? Is it autosomal recessive? Is it autosomal dominant? How does the disease affect the cell? What is the specific mechanism? Which proteins are involved? Which genes are upregulated or downregulated? So essentially, asking these sorts of questions and learning as much as we possibly can about the disease is going to make developing treatments a lot easier. Another thing that we can do is develop molecular drugs to treat the disease. So we're talking about the pharmaceutical industry here, either through natural products, which we've talked about before, combinatorial chemistry, which we're going to talk about later, basically taking already existing molecules and making them better, or by somehow genetic engineering, which we have obviously talked about a lot in the past. This will also include vaccine development and gene therapies, so obviously here we're focusing on disease prevention. Developing diagnostic technologies. It's definitely good to know when someone has a disease versus when they don't. It's always important to be able to divide people into two different groups, the healthy people and the diseased people, so that we can focus on treating the diseased people. So this will involve things like genetic screening, which we'll talk about towards the tail end of this chapter, imaging techniques, which we've sort of touched on before, infection diagnostics. So in this day and age, we're talking about things like a COVID-19 test, wanting to know whether someone actually is infected with a foreign pathogen or whether they are not. Developing devices or machines to assist in diagnosis or treatment. So we're talking about things like respirators, MRI machines, anything that involves technology needed to either diagnose or treat a patient are things that we could focus on if we were a medical biotechnology company. So let's talk about drugs. So a drug is really something that has different definitions depending on whether you're talking about it on a medical basis or a recreational basis. And we are not talking about it on a recreational basis. Medically speaking, a drug is just some molecule or other article that is not food that will affect cellular function in some way for the purposes of altering a disease-causing mechanism and hopefully treating the disease. So a drug is usually going to have its effect on proteins in or on the cell or some other molecules. So some drugs that we are aware of actually have wide-reaching systemic effects, such as what aspirin does. So aspirin, we understand, relieves pain, reduces inflammation, brings fever down. But most pharmaceutical drugs, as we know them, are going to be targeted at one very specific component of a disease-causing pathway. So, for example, infection with the HIV virus, which you can see over here on the right, the HIV virus will infect cells by binding to very specific cell surface proteins. So a lot of antiviral drugs that are targeted at the HIV virus are going to seek to block interaction between the virus and these cell surface proteins to therefore prevent infection by making sure that the one event that has to occur for infection to occur does not occur. But when we're talking about developing drugs, you can imagine that it's going to be kind of a difficult task. So not only do we have to find and develop drugs that not only work, but we need to make sure that they're not harmful. So, for example, if you're infected with bacteria, you can kill those bacteria with bleach, but obviously you don't want to drink bleach because that's not only going to kill the bacteria, but it's also going to kill you, right? So this is the challenge with developing drugs. We want those drugs to alleviate the disease while also preserving the healthy and good function of the organism we are meant to treat. So developing drugs can happen in a couple of different ways. The optimal way to do it is to use what is called rational design. Basically, rational design is possible in cases in which we already have a great deal of information about the disease-causing pathway. We know exactly what the proteins look like, we know exactly what they do, and based on having all of this prior knowledge, we can design the molecular structure of the drug so that it fits perfectly in terms of 3 dimensions and everything else. Based on how much we know about the drug target, which is usually going to be a protein, we can design a drug to do the job exactly. But in most cases, we're a little bit more in the dark. We don't quite have that depth of knowledge about particular proteins or pathways or even the pathogenesis of the disease in terms of how it actually causes a breakdown in body function, so we don't usually have the luxury of using rational design. So the next best thing is to use what is called drug screening. Basically, this is kind of like a shotgun approach. So what we do is that we assemble a library of potential drugs, maybe hundreds or thousands of potential drugs. We don't know if they're going to work, we don't know if they're not going to work, but we're going to try them all. So in order to do something like this, you need a way of doing high throughput screening, which we'll define high throughput here in just a minute. So if we assemble a library of potential drugs, we can test them all for efficacy against the pathogen or disease causer in question. We've talked about this sort of thing before. As you can see in the picture on the right, these discs are placed on a LB agar plate that is growing bacteria, and we can definitely see which one of the antibiotics that soaks the disc is most effective at blocking or inhibiting bacterial growth. So the study of actually looking at drugs and the effects they have on body processes and disease mechanisms, this whole discipline is called pharmacology. So pharmacology is concerned with things like not only the structure of the drug, how well or how tightly it binds to a specific receptor inside the body, determining the correct dosages, determining toxicity, clearance of the drug from the body, how soluble is it in blood and other bodily fluids. So those are the sorts of things that pharmacologists are concerned with. So if you are in the business of developing a drug for treating a particular disease, you're going to want to make sure that you do your due diligence on the pharmacology aspect, because those sorts of pharmacologic screenings are necessary before a drug can get approved by the FDA, as we have talked about before. Alright, so let's round out this discussion by reintroducing the idea of natural products. Natural products are just drugs that are made naturally by cells, and they occur naturally in nature, as the name suggests. So one example that we have not introduced before is a drug called digitalis. Digitalis is a drug that is made by the cells of the foxglove plant, which you can see right here. Digitalis is very useful because it can be used to treat a number of different heart conditions like arrhythmias, problems with electrical conduction in the heart, and then also problems with getting blood to flow from chamber to chamber as well. So this is very good and very interesting, but one of the problems is that naturally occurring drugs can't satisfy all of our medical needs. There's not a plant to cure every disease out there. So oftentimes what researchers and scientists will have to do is take already existing natural products and improve on them through a process called combinatorial chemistry, which is going to be the focus of our next video. So thanks again for your attention, and I will see you next time.